Okay, good to go. So, as y'all know, there's an external, a middle, and an outer ear. The external is just basically everything you see and everything you put a Q-tip in and you can touch with a Q-tip. Okay, if you've hit the middle ear, you've gone too far with that Q-tip. Okay, you could actually rupture your tympanic membrane. Kids stick stuff in their ears all the time, like sharp stuff even, because kids are dumb. We were all kids, so we were all dumb. Middle ear starts with the tympanic membrane. You don't want to rupture that because it's very painful. There's a lot of nerves in the tympanic membrane. There's actually one running straight across, a pretty big nerve running right across the tympanic membrane. So you know if you're in there, you're in the middle ear. Of course, the inner ear is dedicated to nervous system stuff, so balance and hearing. Do you guys have an okay time with special senses, Learn Smart? That was all right. I feel like you learned a lot from that Learn Smart, or was it just like
the one we just did?
Decent? Yeah, that was the one you did last week. No thoughts
I
on
feel like
that?
I'm getting confused
Don't
between
remember that?
general senses and special senses.
I'm sorry?
I get getting confused between general and special.
General and special, okay. Yeah, we can go through that right after we're done with the special senses, and we'll do a quick review on that. Just remind me before break. Okay. Outer ear. In fact, every single one of these folds has a name. You know, all these little, you see all these little ridges inside your ear, and they're all uniform. We all actually have the same ones, as long as your ear developed properly. The only thing that's different about ears is some people have attached earlobes and some people don't. That's a skin thing, not necessarily an ear thing. Okay. So this is called the helix. This is the anti-helix. Okay. This is, together they make the auricle. The little thing that holds in your earbud, your earbuds. Can you guys see that? You touch that little thing. It's called the tragus. Okay, that's a Jeopardy. It's maybe a Jeopardy answer. What holds in your earbuds? Okay, you shouldn't wear earbuds, by the way. Wear the loose ones that just sit in your ear. Okay, you'll get less hearing damage that way. Millennials are going to have some issues with hearing damage for sure. I know I will. I mean, I always wear those things. Used to. The tympanic membrane, of course, separates the auricle and the external acoustic meatus, which you already learned in skeletal system. Those two are part of the external ear. So remember that the tympanic membrane is like a drum. Okay, so vibrations, the sound waves cause vibrations of this drum and project them into the middle of the ear right here. So middle ear is consisted of what's called the tympanic cavity, which is basically this box, this spacious box that holds the ossicles, and also, of course, begins at that tympanic membrane, carries vibrations from the membrane to what's called the oval window. The oval window is the entrance to the inner ear. So if you're at the oval window, if you were a vibration and you traveled through the ear bones in order, how would you travel through those bones? How would you list those ear bones in order? Sabrina, go ahead. Malleus, Incus, Stapes. We know those as, gosh, I can't even remember what the layman's terms are. The mallet, the anvil, and the hammer, the anvil, and the stapes, or something like that. I don't remember the layman's terms. Are they on there? Okay, very good. Hammer, anvil, stirrup. The stirrup looks like a stirrup if anybody rides horses. So if you're a vibration and you get through the stirrup and you hit this oval window, you know that you're about to go into the cochlea. These signals don't go into the semicircular canals. Those are for what? Balance and equilibrium. These signals specifically get shuttled into the cochlea. Okay. So I want to show you a more detailed picture of the middle ear. This is the malleus here. These ear bones are held in place by little tiny ligaments, of course. And once you get to the end of the ear bones, the stapes, the base of the stapes looks like an oval. Okay, so therefore it's attached to the oval window, which is just the little window that holds the stapes in place. If you're at a Lady Gaga concert and the music is really loud, anybody been to a Lady Gaga concert? I feel like it'd be a weird time. Don't you think so? That would be really weird. She's a weird person. But you know, if you're at that Gaga concert, you're right in front of the speaker, or a clips or something like that. This is going to vibrate too much. So too many sound waves go in, cause too heavy of vibrations here, which will damage that signal once it gets into the inner ear, damage the inner ear structures. So you have dampeners. Two muscles in the body, there's the two smallest skeletal muscles, are called the stapedius and the tensor tympani. And they attach to what's in the name. Okay, so stapedius muscle attaches to the stapes and will constrict and dampen, kind of like if you put, if anybody's ever played drums before, or even like you can just visualize putting a rag on top of the drum and then hitting it, and it's going to really dampen that sound. That's what these muscles do. The tensor tympani, excuse me, the tensor tympani actually attaches to the tympanic membrane and holds it tight so that those sounds can't even necessarily get in there. Sabrina, go ahead.
You answered my question, but I guess I can come up with another one.
Okay.
As a person ages, does the tensor tympani become weaker or like loses its elasticity?
Yes, because every skeletal muscle does atrophy as you get older, so therefore these would too. So that's why, you know, it's another reason that we get hearing damage as we age, other than the earbuds and the concerts. Right? We get some permanent damage in there. So let's talk about otitis media. I want you guys to write down otitis media, because that is middle ear infection. So you may have heard of this before. It's also called swimmer's ear. Okay, so otitis media is, as the medical term suggests, itis, the inflammation. Media is middle ear. How do we get otitis media? That strep or whatever is in the throat, whatever harmful pathogen, travels up through the eustachian tube. So there is a connection between the middle ear and the throat. Why? Why do we have that? Yeah, so that when you go up in a plane, or you go down a huge hill, say on a road trip, and you go down this like steep grade, the pressure is either going to go up or down in the ear, and it's going to want to pop. Okay, so what releases that extra air, if you will, is the eustachian tube. So it's like a release valve for pressure in the ear, and that way, you know, you don't have to, you know, your middle ear won't explode. Is basically what it's helping you avoid. Why do kids get these otitis media infections more often?
So the eustachian tube really is inflamed, so it wants to drain.
Almost. It's not that it's a little bit more constricted in a kid, so it won't drain as properly, but it has something to do with the orientation of the tube. That's part of it. It's a little bit shorter, and it's placed a little bit more laterally or horizontally. So it would be the difference if you're a strep bacteria and you have to climb up a really steep, tall slide. That would be difficult. But if you just have to run across a slide, right, from one side to the other, that's going to be easier. So it tells you it's easier for infections to get over there, just physically, distance-wise. Okay, so once you get into the inner ear, you're going to see this sort of setup. Here is the round window, which is the exit point of signals getting into the ear. The entrance point is the oval window. So what happens is a physical vibration of the bone, those middle ear bones. It's like Morse code. Think of the stapes, the last ear bone, is Morse code. So actually physically tapping on this bony labyrinth. Those physical tappings, this whole structure is filled with fluid. So if you tap on the bone bone surrounding that fluid, it's going to shift the fluid around inside it. So the stronger the tap, the stronger the wave through it. Okay, it would just be like there's a pool of water and you smack your hand against the top of the water, that vibration will travel further. Okay, so those fluid shifts are actually happening inside the membranous labyrinth. Okay, that's the innermost. See the purple layer here? That's the membranous labyrinth. So those signals are shuttled through here. The stronger the tap, the farther that signal will travel down the cochlea. Those are going to be the lower frequencies, get further into the cochlea because they're going to produce the strongest waves. Okay. So we'll talk about that in just a bit though. Our anatomy, the cochlea, which was on the semicircular canals was on the lab practical. We're on the lab practical. And then you've got the vestibule, which is this bulb in the middle. And for some reason, it's easy for me to remember. Vestibule is a bulb. I don't know if it has something to do with that last part of the word, but that's the bigger part. That's the bulbous area. And then you draw a division line here, and that divides the equilibrium portion of the ear from the hearing portion of the ear, which is the cochlea's job. So those are the three parts of the inner ear. Two of them are responsible for balance, equilibrium, and one is responsible for auditory signals. And they both pass their information through what cranial nerve? First, give me the number and then give me the name. Yeah. What's the number?
The number is 5.
Nope.
6. 7.
One more.
It's the vagus nerve.
Nope.
No, it's the vestibular cochlea.
Vestibulocochlear. The other V. Yeah, there are actually a few V and a few G ones, so it's easy to get those confused. But it's vestibulocochlear, which if you have a mnemonic, you can work your way through that and get to cranial nerve.
How's the name of the structure though? Vestibular.
Yeah, and that's why I made the point, is because the name of the nerve is related to the anatomy that conveys that information. So that one is actually one of the easier ones to remember in terms of naming. Remember I said the name tells you a lot about it. Okay, the cochlea is snail-shaped, and it's shaped like a conch shell. So a conch shell, snails actually inhabit shells, okay? So they can exchange, and get a shell exchange if they want to go. I feel like a huge animal, like a whale, maybe like a dolphin. Some animal would have the proper size cochlea for a snail to invade. I don't know which one it would be. There's probably some snail has found it on the cochlea. I'm sure if it's found it on the ocean floor. Wait a minute. The dolphins have cochlea? Yeah, they would. Yeah, they've got all the same cranial nerves. I don't know crap about animal anatomy. I assume it's all the same, right? No. Horses have 4 stomachs. That's the only thing I know. Okay. So this shelf here in the middle is the spiral lamina, and the shelf in the middle is the modiolus. It's the core, and then these dividers here are called the spiral lamina. That nerve is getting impulses from all the different parts of that tube. So this is just that tube coiled on itself. The windows, so you want to remember, okay, so O comes before R in the alphabet. The oval window is the entrance for fluid signals, for vibrations. The round window is like actually the exit. So that's one way to remember that. O comes before R in the alphabet. The oval window comes before the round window in terms of flow of auditory signals. And actually the only purpose of the round window is to allow excess fluid shifts out, excess vibrations to be released out of the ear. So it's kind of a release valve as well, kind of like the auditory tube here. Okay, Stapes knocks on the oval window. What happens next? That oval window passes that vibration. Remember, now it's a fluid vibration. Okay, so you tapped on a wall that was beyond the wall was all fluid. Okay, so now that fluid vibration is going to be passed through. You have these scalas that you studied in the inner ear. That's going to first pass through the scala vestibuli. Okay, so those fluid signals, if you unrolled the cochlea, would travel through the top, excuse me, the top space first, and then if that signal is strong enough, it's actually going to coil around the end, and then come back through the scala tympani, which is on the bottom. So if you're a really strong fluid shift, you're gonna go all the way over, okay, and you might even come out the round window. If you're a really a low volume fluid shift, you might get to here and stop. Remember that the receptors for hearing are located inside this scala media, which is the really small duct. Okay, what happens is there is a set of receptors ranging from high frequency receptors to low frequency receptors, and those detect those fluid shifts. So wherever the fluid shift ends, that receptor picks up the signal. Okay, it's just like kind of like shuffleboard, right? So like shuffleboard, you get it further down, and in the right area, you get that amount of points. That's kind of how your cochlea works. It's the only way. That's how I think about it. Think about it as one big shuffleboard inside the cochlea. Okay, so that's where the action is there in the cochlear duct. And I want to show you a zoomed in view of that from a cross section. Signals enter through the vestibuli up top, and then you have a vestibular membrane. Okay, that vestibular membrane separates the scala vestibuli from the media, which don't get confused. You know, in anatomy, you can get confused if you don't know what terms can be interchanged, right? So that's also good. The cochlear duct is also the scala media. Those two things are the same. So you might want to make a note to yourself. See cochlear duct, that's also the scala media.
So we could write both on the exam.
And either or would be just fine.
Yeah.
Coffee's disgusting. Don't get coffee at Square Donuts. I took a risk and didn't pay up. He gave it to me free though.
Yeah.
Oh, she's like, this batch is bad. He'll be unhappy if he pays for this. Square Donuts, yeah. Yeah. The strip mall looked pretty shady, no lie.
Yeah.
Okay. So the specific membrane or the roof that covers that scala media and those hair cells, you see those receptors here. It's called the tectorial membrane. Tectorial plates are plates in the earth that shift around, so the tectorial membrane is shifting and then irritating, stimulating those receptors based upon how strong the signal is. And then below it, what is the basement or the floor for those hair cells is the basilar membrane. So think basilar. Anytime you see basal, remember that's the basement or like lowest level. So there are 3 ducts, 3 membranes. Each has their job. This is the entrance duct. This is the exit duct. And this is sort of the receptor duct. Okay. Any questions so far? So we're just kind of like zooming in more and more, looking at the details here. So when you look at just this area, so now we zoomed in even more, and we zoomed in on the tectorial membrane with those cells underneath it and the basilar membrane. So we're seeing this thing. This is called the spiral organ because it spirals around the cochlea. Okay, and it is an organ. It has a specific function that is to detect sound. We talked about cilia the first week. We saw them in the olfactory epithelium. We see them a lot in special senses. You even saw them, well, those were microvilli in the taste buds. You You see cilia because they're a little bit larger in hearing cells. You see those here in the spiral organ on top of the receptors. So that makes sense though, right? Because cilia are kind of like these little fingers and they're very sensitive. So as soon as they experience that fluid shift, okay, they're kind of like buoys, right? As soon as they experience that fluid shift, and buoys are in the lakes and the oceans and stuff, and they detect fluid shifts too. As soon as those little buoys start to wave, they fire their action potentials back through what cranial nerve? Vestibulocochlear, but specifically the cochlear part. Right? I'm sorry. Yes, the cochlear part. I'm sorry. Go ahead.
Like it's a sensory nerve.
It is a sensory nerve, yeah. So it's a peripheral sensory nerve, specifically going through the cochlear portion of vestibulocochlear nerve. Remember that that's just like a bundled nerve. It's carrying two separate signals, but they travel together in a bundle. It's like a package deal. Those cilia are given a specific name, stereocilia. Your stereo produces sound. It's pretty clever, right? Good job, anatomists, early anatomists. Okay. You know, I feel like they weren't too creative. All they could come up with was either their name to name something, or Latin, Greek or Latin.
They got tired by the time they got to the inner ears.
Yeah, they were probably super bored by that point. So hair cells work like buoys again. These are the stereocilia. So this is an actual picture of those stereocilia, and they look like cilia, made of microtubules. This is an actual image which actually resembles the diagram pretty well. Pectoral membrane on top, maybe T for top, and basilar B for bottom. By the way, that's the only way you guys are going to remember this stuff eventually. Okay, like stupid stuff like that. Just one letter is all you need. That's really, I mean, when I'm teaching, if I didn't see this, that's how I would remember pectoral top, basilar bottom. This is the kind of like piano keys. I like to also think of the scala median, the spiral organ is like one big curled up piano. So when you uncurl it or when you unroll it, you're going to see the higher frequency receptors towards the left side of all those piano keys over here are going to be high frequency receptors producing high sounds. And the piano keys down here are going to be low frequency receptors detecting low sounds. So if anybody plays the piano, you kind of know that, if we're all playing it together, you kind of know that the low keys are on the left side and the high keys are on the right side. So it's like a flipped piano, okay? It's upside down. So it's not upside down, it's just flipped around, it's turned around. And this is how your brain knows what frequency signals are coming in, guys. So you have the auditory cortex, which is located in what lobe?
Temporal.
The temporal, right? That one's easy because the temporal lobe is right under the ear. So actually, the vestibulocochlear nerve is kind of interesting because it doesn't have to go through the relay center in the brain, the sensory relay center, grand central station, which is what brain structure? The thalamus. Remember, that's where all sensory signals go and are then distributed to the proper areas in the cortex. This one actually bypasses the thalamus. It's strange in that way. It's novel in that way, is that it doesn't need to go there. It actually just the signal is taken straight to the auditory cortex. So in the auditory cortex, you have a strip of land or a strip of cortex. Some of that strip of cortex, it's laid out just like this, right? You have cortex dedicated to, deciphering these impulses, and then these here. Now you're kind of building the picture. Okay, somebody's having a seizure, extra electrical activity in the cortex. In the area that's dedicated to the higher sounds, they're going to hear like a higher sound of an auditory hallucination if it's starts somewhere over here in the auditory cortex, and you're gonna hear like a whooshing sound. And now you can kind of say, a lot of people say they have whooshing sounds for their aura. Have you ever heard this? If you talk to like somebody with temporal lobe epilepsy, they'll say it's like a rushing, sounds like rushing water starting low and going high. So now you can kind of know why, right? Stimulating low cortex going to high. I know that's a little tangent, but I think it's interesting. It helps us kind of understand the relationship of brain to your perception. Okay. Any questions on how an auditory signal gets to the brain? I want you guys to work through, okay, so picture just a sound wave and then trace its path through the anatomy of the ear to the cortex. Remember, it doesn't have to go through the thalamus, so you don't have to put that part in there. Straight from the cranial nerve to the cortex. I want you to write that out for yourself. You can use this as a guide, but try to do it through memory with your neighbor. So start talking to your neighbor and construct this pathway. Sure.
So like, what about sound waves are? Like, so say without, I get without. So it never really has like,
Yeah, tinnitus. Did you ever experience any sort of like damage, like hearing damage from any loud sounds or anything? Oh. How's that related?
How do you hear that?
Yeah, so that's not your cortex being stimulated. Those are your hair cells here. Loud signals actually start to knock out hair cells. Like say you're experiencing like hearing damage on the receptor level, which I have too. I've been around my nose, so I was always around that stuff. And these cells get damaged, and for some reason they respond to damage by turning up their response. So like that just means those sounds, that frequency of sound, is telling you what receptor is freaking out due to damage. Yeah, so it's actually an actual potential. It's a memory, but it's an improper one. It's coming from a damaged hair cell. Most animals can actually regenerate their hair cells, but humans cannot. It's a weird thing. That I do know that about animal anatomy. Most other mammals can. These other cells will differentiate into hair cells when these are gone, but we can't. We're stuck. And so, yeah, just gotta protect your ears, earbuds. Not earbuds, but the foam things to stick in your ears with contacts or earbuds. intense detail, like that connection between the sensory receptor and then the actual sensory nerve that's carrying that impulse. It's like a two-step. It's like the hair cell is just an accessory, and then it releases some neurotransmitter that stimulates the actual nerve going back to the brain. So you think of it like a two-step.
And then that stimulates, you know, and that's how you get to higher level.
So a good way to study this would be to have one sheet of paper divided into two sides. On one side, write out the words, the flowchart that you just did, and then on the other side, draw out an actual picture of the signals going through. It's not going to be pretty, probably, but that's okay. It doesn't matter. This actually, in fact, the simpler the better, as long as everything is labeled. So notice between steps 10 and 11, between the cranial nerve and the temporal lobe, there's no thalamus listed. So that means it doesn't have to go through the thalamus to get there. It goes straight there. Which makes sense because that would take longer, right? We want to get that signal immediately. Okay, next is equilibrium. Excuse me. Equilibrium is detected by the other side of the ear. So the division line is right there at the base of the cochlea. Now you have two parts of the inner ear that we talked about, the vestibule, which detects static equilibrium. So that means when the body is not moving, it can sense the position of the head. So while you're sitting down, if you close your eyes, go ahead and do this, and move your head around, you know where your head's at because your vestibule is interpreting those fluid shifts. Okay? You have no visual input. Now, dynamic equilibrium senses rotation and movement of the head and the body. So that means if your body is moving, now it's not the vestibule's job to detect those fluid shifts, specifically acceleration and deceleration. Think about a car. If you close your eyes and you were stationary in a car, okay, picture yourself in your car. Put yourself in your car. Okay, now you're sitting there, you're completely still, unless you get road rage. And then you're moving forwards and you're moving backwards in space, but your body's not moving. The way you know that is fluid shifts inside your semicircular canals. Then those can detect that. Okay, so we've looked at the utricle and the saccule. Those two are those are the two major portions of the vestibule, and they are listed in green. They look like green jelly. Okay, those are your utricle at the top, upper, utricle upper, and saccule below. Each has a macula, okay, which is responsible for, it's like just a group of cells responsible for detecting that motion. Same thing as the other ones. They're hair cells with cilia. Okay. So, excuse me, when, this is really, really cool. I mean, these details are really interesting to me. You have a bed. Okay, so here is the utricle, and there's a bed of cells. Okay, a nice pillow top mattress, kind of firm from the support cells, but still, you know, a little bit soft. So you don't have to be sore in the morning. You've got hair cells here with their cilia, and then you just have a blob of jelly filled with rocks. Rocks. I'm serious. There's just little tiny rocks in your ear. And they're called otoliths. Lith is a medical term that means what? Do you know what a monolith is in terms of building?
It's just a big block.
It's a big stone building. So lith is your word for stone. So these are tiny stones. Odo, ear stones. Isn't that cool? That tells you they're in the ear. As your head moves forward, the stones shift the jelly. This is so stupid. The stones shift the jelly around, which stimulates those hair cells because they bend. So depending on the direction the hair cells bend, okay, they can tell your brain what direction your head's moving when your eyes are shut out. So the rocks move the jelly bends the hair. You know what you're doing. Yeah.
Okay, we were talking about rocks. Does anyone know what their constitution is?
Yeah, they're really hard salts. Yeah. Yeah. So like on the periodic table, I'm not sure what their actual consistency is, what kind of salts make them up. They would be like the hard salts somewhere in the alkaline metals in that area. Yeah, I'll have to look that up. I'd like to know. I'd ask that question. I'll figure it out.
Hard salt,
Yeah.
I think it's like a salt metal, something like that.
Yeah.
Not silicone.
Yeah, that's too soft. It's got to be something harder. So hard enough to where is like when these rocks shift around, they can move the jelly. And when the jelly moves, that stimulates the hair cells in that direction. Now these rocks can break off. It's gonna get sore. I have my side. These rocks can break out of their jelly prison, and they can roll around the ear, and that's what causes vertigo. Is they stimulate hair cells that shouldn't be stimulated. So you think you're moving when you're still. Rogue stones traveling through the canals will cause some serious issues. Do you know anybody with vertigo?
I get vertigo.
Yeah. Now, when you're younger, it's not always due to this rogue otoliths, but when you're older, that's what happens. They get loosened up, they travel through your ear, and it's debilitating. There is a home remedy for it, and it works. I'll tell you about that in a second. Joe.
How do those things get there? So do they just?
I don't know. That's a good question.
And
I mean,
why?
Okay, so yeah, you're making me think about embryology. Every cell in the body differentiates from a stem cell, right?
Mhm.
How can we get from cell to stone? I don't know. It must be metals and salts that accumulate in the bloodstream during fetal development that are, man, I don't know. That's really cool.
It is
That's a cool question. I'm gonna have to figure that out too. I don't know that.
like to wrap your head around that. I can't really wrap my head around how
I
in
can't
the
either.
world do we have rocks in our ear?
That's why I'm so blown away by this concept. I'm sorry.
Is it like a kidney stone, like the makeup of it?
So that's the thing is when we talk about gallstones, kidney stones, those are just concentrated salts.
Mhm.
So there's got to be some process that your body does during development
Right.
that takes the salts inside your body that you've consumed or that are in mom's bloodstream and just arranges them into these. Because that blows my mind that your body's that specific and can do that. Because it has to happen.
Yeah.
We just I don't know how. I'm sure it has something to do with
Right, with embryonic development and
Yeah.
everything.
If you don't know how something works in the body, blame it on embryology. That's what my professor used to say. Okay, so vertigo. Write this down. The half somersault. I'm not talking about gymnastics. It's a neurologist that came up with a home remedy to treat vertigo. If somebody you know or if you experience vertigo and it's from these, if you follow those directions and those motions, it's like a special yoga session. You move your body around, and if you follow those directions precisely, it will probably cure your vertigo. Pretty amazing. I'd show it in class if we have time. Maybe I'll show it at the end. We don't get these. Okay, so remember that these impulses are going through the vestibular portion of cranial nerve. Excuse me. Okay, so we've got the vestibule. Both the utricle and the saccule work in that manner with the rocks, stimulating hair cells. If you know the mechanism of action there, instead of fluid shifting around the hair cells, it's rocks shifting around jelly that shift around the hair cells. The same exact mechanism happens in the semicircular canals. So these are called the ampulla. These little bulgy areas at the base of every canal. And they are little swellings that also contain jelly. When the jelly shifts, it stimulates these little nerves. So, excuse me, we're going to look at the crista ankularis, which is the sensory organs at the base, the ampulla, that actually conduct those dynamic equilibrium signals. So when your head and body are moving. At the base of every, I know it's a lot of terminology, guys, but at the base of all the crista ankularii, you have a little cupula. And that's the jelly. That's where you find the jelly. So one more time. Inside the semicircular canal, at the base, you find an ampulla. There's a smaller area inside every crista ankularis that contains the jelly. And that's the cupula. So, cup of jelly. Alright, here's, it looks like actually a little flame. Little tiny flames at the base of every canal. This is the crista ankularis, and this is the cupula portion of the crista ankularis, which contains the hair cells. And these cilia are long. These are actually longer cilia. You can actually see that in the picture, right? They're a little bit longer. There is fluid circulating in this space. Okay, so you need to visualize your head moving forward. The fluid shifts inside that semicircular canal. As the fluid passes by the cupula, it pushes it to the side. Okay, so fluid is rotating around that canal, pushing on the cupula, irritating the cilia, which produce nerve signals. It works in the same way as the cochlea hair cells and the utricle and saccule hair cells. So these are called, what kind of receptor sends impulses due to physical disturbances? What kind of receptor is that? What classification? I'm sorry? Not the type of nerve.
General?
The type of receptor. These are special senses.
Oh, special, right, okay.
Remember, so let me spark your brain. The receptors that detect chemicals in the nose are chemoreceptors. The receptors that detect light signals are. So what are these receptors? Mechanoreceptors. Like a mechanical arm is an arm that moves, physically moves. Okay, so mechanoreceptors detect mechanical shifts. This really small picture kind of does it for me, and I apologize that it's a small line on the textbook. As the fluid shifts through and passes by the cupula, you can kind of tell that it would shift it around like a buoy, okay, and then it fires in response to that. So it's the same mechanism. Instead of fluid, though, instead of fluid directly stimulating those hair cells, it's jelly in the vestibule, okay, in the balanced part. So add jelly, and you get the equilibrium part of the ear. Okay. Really quickly, I want you guys to do the same thing you did for cochlea, for hearing, but just do it for one of the two, either the semicircular canals, how a fluid shift turns into a balanced signal from start to finish. Most of the steps are going to be the same. Raise your hand if you've got questions.